Thank you so much, Roger. Thanks. It's really a pleasure to be here. And what I want to do today is to review with you how the large observational cohort study, prospective cohort study, really support our clinical trials endeavor. When we have an epidemic, usually the first thing that we do is to try and observe the disease, and how does that whole experience really support advanced clinical trial effort. So these are my objectives: to give you a little bit of a historic perspective from previous epidemic, particularly the HIV epidemic, and then talk about the RECOVER observational cohort study, particularly the adult cohort that I chair, and then some of the lessons that we have learned from RECOVER and how they apply or aid ongoing clinical trials of long COVID. So why do we do observational studies? One, to characterize the disease and its associated conditions, to estimate disease burden, risk factor, and complications, to understand the trajectory of progression or recovery, and also to understand pathobiology and biomarkers discovery, and then ultimately to find accurate diagnostic tests and biological target for treatment and prevention. So, three major things. We want to, first of all, estimate how common the burden of the disease. Then two, we want to identify, define the cases. How do you really identify a case, define them? Then the biomarker for treatment, the biomarker for progression, and the biomarkers for diagnosis. So those are some of the things that we really think about as we continue to follow our observational cohort study. So this is the first cohort that I just wanted to highlight. This is the MARCS cohort that was started in the US when the HIV epidemic started. So the MARCS cohort is mainly men, especially men who have sex with men, was established in 1984, just shortly after the beginning of the HIV epidemic. Here I am highlighting a key finding from the MARCS cohort. So it was in the MARCS cohort that we discovered that CCR5, the co-receptor that is used by HIV for gaining entrance into the cell, that there are people that have mutation in the gene that encodes for this particular receptor. So if you are homozygous for that gene, you are almost resistant to HIV. And if you are heterozygous, if you have HIV, you are a slow progressor. So this observation came out of the MARCS cohort, very early in the course of the MARCS cohort. And that observation led to so many things. First of all, the discovery of Moraviroc, which is a drug that is used for treating HIV. That observation also is kind of really the basis for a lot of the cure agenda today for HIV infection. We know there are 7 people that have been known, recorded history of HIV cure in the world. And those 7 people came out of the observation, this particular observation that came out of the MARCS-wise combined cohort study, that if you are homozygous to CCR5 mutation, you don't develop HIV. And these 7 people that have been cured, what happened was that they had cancer and they underwent bone marrow transplant from donor that were homozygous for this particular gene. So here is another cohort study. This is the HIV outpatient cohort study funded by the US CDC. My friend Frank Palella from Northwestern University, early in the course of the AIDS epidemic, once treatment came in 1995, we started introducing antiretroviral therapy. Frank noticed, using the HOPS cohort, that if you start people on antiretroviral therapy, mortality goes down. Not only that, the incidence of opportunistic infection significantly decreased. And what did that lead to? That changed the way we treated opportunistic infection in HIV-infected individuals. All of the prophylaxis, guidelines for prophylaxing opportunistic infection changed because of this observation from the HOPS cohort. So the last cohort I'm going to mention before I go into COVID is the WISE cohort. So this is the Women Interagency HIV study that was also started at the beginning of the epidemic in 1993. So this was this is a study that was led by Mash Cohen from Rush University in Chicago. They observed that while people were doing well with antiretroviral therapy, mortality rate went down, but it was still higher than what you see for the general public. And what came out of that was that people started looking at why is mortality rate higher in people with HIV that are treated, successfully treated, they have suppressed viral load, CD4 cell count came up. So that led to a series of studies. And that series of studies led to the big REPRIEVE study in the United States that was published, was it last year or 2 years ago, using statin for prevention of cardiovascular disease in people living with HIV. So these are some of the power of observational study. You observe things that then is taken into clinical trials. So the pandemic came, and then not long after the beginning of the pandemic, we began to notice that people were having long COVID, long hauler. And not only, everybody had brain fog from the pandemic, not just, we all were so exhausted from the impact of the pandemic. And so that was when the NIH put together Recover. And that people are recovering from the acute phase, and they are not recovering completely. And so at the time Recover was established, there were so many gaps about long COVID. One, a lot of the published paper studies at that time, this was towards the beginning of 2021, a lot of the studies were dependent on electronic health record or self-reported e-cohorts. And there were gaps in these studies. One, it was the population that was impacted was underrepresented. Reliance on self-report. The sample size was small in some of the studies. Some of them were single site. If they were multiple site, they were limited multiple site. And then at that time, they were very limited follow-up, maybe 6 months of follow-up, a year of follow-up. And then, of course, we didn't have a definition of what long COVID was. So long COVID, even till today, is still what you want it to be. We have different definitions. We have the Recover Index. We have the NICIM definition, the WHO definition, CDC definition. There's really no consensus on what the definition. And, of course, we didn't have any single drug for treating long COVID, and we still don't have a single drug that is dedicated for treatment of long COVID. So when Recover was started, we had 5 goals. How many people are getting long COVID? What are the symptoms? And how long does the symptom last? Why do people have long COVID? And can we find a way to treat or to prevent long COVID? Essentially, the question is the disease burden, the disease severity, the trajectory of disease, pathogenesis, and target for treatment. So this is how Recover was configured, for those of you who are not familiar with Recover. It was a big study, so many components. You have the clinical science core, the data core, biorepository core, you have the cohorts. So you have the adult cohort, the pediatric cohort, the autopsy cohort, have electronic record, and then you have a lot of data resource, imaging, clinical, pathology. It was a very, very big study. And so these are some of the PIs of the adult cohort. I think Sally is somewhere there. Sally is a PI along with me in the adult cohort. There were 15 hubs across the country, 86 sites all over 33 states and 83 sites, including DC and Puerto Rico. So the design was what we describe as an ambidirectional longitudinal meta-cohort. So ambidirectional, we followed people prospectively. We also collected data from them retrospectively. Meta-cohort, because there were so many cohorts. You have the adult, pediatrics, pregnancy, as well as the pediatric congenital cohorts. So this is how the study was scheduled initially. So the first year, people were seen every 3 months. We collected data, and we also collected samples, extensive sampling. After the first year, we collected data from them, survey every 3 months, but they came in once a year for sampling, for sample collection. So when we were done with enrollment, about almost a year ago, October of last year, we had enrolled into the adult cohort almost 15,000 individuals. And so these were enrolled in these 4 buckets. So we have people who were enrolled in the acute phase, that is within 30 days of their acute COVID-19 infection. And then people, the post-acute phase, which was after 30 days. And we also had the uninfected control at that time. And so all of that together was about 15,000. So 33% of them were acute, 50% were post-acute, and about 17% were control. We also were very careful to make sure that we enroll people in a way that it reflects the demography of the United States. We wanted this study to be representative. Whatever came out of it can be can be generalizable. And so these are kind of where we are in terms of what we have collected so far. Tons of serum, tons of plasma, tons of PBMC, nasal swab, urine, saliva, stool, vaccine, WBC, and then the clinical and demographic information that were collected are on the other side of the slides. So we also did a number of specialized procedures. So we call them tier procedure, tier 2 and tier 3. So these include a list of things that are listed on the slide, things like oral glucose tolerance test, ACTH, cortisol, Hep B, Hep C, 6 minutes walk, chest CT, fibroscan, on and on and on and on. Then in terms of a tier 3 test, we did more specialized procedures, such as on the other side, radiologic studies, brain MRI. We had CPET, colonoscopy, bronchoscopy are being rolled out. So it's a really extensive characterization of this population. And so what are the lessons that we learned, and how are they now supporting some of the ongoing clinical trials? The first question was the question of case definition. How do we come up with a definition that is acceptable, that can actually be objectively used in clinical trials? Because you can have a successful clinical trials if you don't have a consistent way, objective way of defining your case. So what we did was that we took, in the first analysis, we took about 10,000 people in recover, some of them with a long history of SARS-CoV-2 exposure, the others without exposure. And then we looked at 44 symptoms that we elicited. Which of them are more common in people that were exposed to SARS-CoV-2 infection? In this simple question, we were able to identify about 33 of them with an odd ratio of 1.5 or adjusted odd ratio of greater than 1.5 that were more common in people that had a SARS-CoV-2 infection. Then using this sophisticated statistical approach, these symptoms were weighted, and then they were scored. And using the lasso approach, which is a kind of a logistic regression, we were able to really define an optimum score that can actually predict somebody that has long COVID. And that score was 12 in this initial exercise. And so this was not really this was a highly specific. So if it calls you have long COVID, you truly have long COVID. But it's not very sensitive. So there were people with like single organ long COVID who were not, who did not meet that score. And then more recently, we did a redefinition of this score, where we added more data, added more participants. It became more sensitive, a little bit still as specific. And so this has now become almost the, what you call the most, the single objective definition of long COVID that is out there. We agree that it picks up only those with very severe cases of long COVID. But when it calls it, it does cause it. And so a lot of clinical trials are now using this index, what we now call recover index, in their studies. The other thing that we were able to do was to identify the different types of clusters. So we know that long COVID is not a homogeneous condition. So this exercise allowed us to identify the different clusters. And we were able to cluster this population into 4 different variants of long COVID. And many of the clinical trials that you see today are designed to really address the symptoms that are common in these variants. For example, the VITAL study, we have the neuro study, we have the sleep studies that is going on within recover. So in the interest of time, I'm just going to run through all of these. We have a lot of studies now, funded mechanistic studies, to actually help us identify biomarkers. Biomarkers that we can use for monitoring disease progression, biomarkers that we can use for treatment target, biomarkers that we can use for diagnosis. So these are some of the studies of biomarkers. The one I wanted to highlight is the WATT study that we published about a month ago that actually showed that the spike protein is persistent up to 12 months in people that have in people, in general, 21% of the entire population has spike protein. And that if you have long COVID, your chance of having persistent spike antigenemia, either the spike component of the spike or the end protein, your chance of having persistent antigenemia was about 2-fold higher compared to those who do not have long COVID. And then there were a series of other studies that are actually looking at different immune profiling of long COVID. And so what we're doing now is that there's a big system biology initiative within Recover. It's just multi-omic approach, looking at it from all dimensions. And once this is done, this data is going to be available to the public. We would know a lot of the immunologic signature that is associated. And we're doing this in, almost all the 15,000 people in the adult cohort, as well as the 15,000 people in the pediatric cohort. We've expanded our analytical capacity. We started what we now call the network of biostatistics to really help us increase the amount of our statistical capacity to really publish some of this work very quickly. So in conclusion, observational studies can provide pertinent information needed to advance clinical trials. One is just consistent repeated measure of the large study set, measure of same large sets of symptoms enable deep understanding of clinical features necessary for case definition. Also, we are able to define the incidence as well as the trajectory of the disease, burden of disease, and severity of disease. The other thing that we've been able to, that this observational study can help you do is really subpopulations. So if it's large enough, you'll be able to look at manifestation in subpopulation. But more importantly, look at phenotypes that can help with personalized therapy. And lastly, of course, target for treatment, diagnosis, and monitoring of disease progression. So thank you so much for your attention.